All right, you guys, it's time for the protein. So proteins are our fourth and final class of biological molecule. They are the busiest. This is probably going to be the longest part of the lecture. I know you've been looking forward to it. Proteins do so very much in our body. There's a lot to say about them because they are just so important in our bodies. Proteins are present in every single cell in every single organism. They control the rate of chemical reactions in something called an enzyme. They form cellular structures. They transport nutrients and waste through the cell, but then also through the body. And they are responsible for fighting disease. So they do how quickly you can survive. They make up what's actually your body. They transport everything that you need, and they keep you healthy. Proteins are so important. Each function of the protein is going to end up being a different structure. One of the big topics in biology is that structure and function go together. Okay, the structure of something is going to tell you a little something about its function. So we're going to be looking at that in just a moment. At its simplest, proteins are made of monomers called amino acids. So we have 26 letters in the alphabet, and from that we can write endless words, sentences, novels, stories, forever, right? Right. Well, proteins have 20 different amino acids that they can use, and these 20 amino acids give way to every kind of protein that does every kind of function in the whole entirety of life. These are the kinds of things that give us evidence for evolution and for common ancestry. Every living thing uses the same 20 amino acids to build their proteins. Every living thing uses the same 4 bases to build all of their DNA. We have such commonality with every living thing, from plant to animal to fungus to protist to bacteria to viruses. We have so much commonality. The structure of an amino acid has an amino group with this nitrogen with the 2 hydrogens, and it has a carboxylic acid group, which is the carbon double bonded to an oxygen to an OH, and an H here. And then the only thing that changes in the 20 amino acids is this R. We sometimes use the letter R. I think of it like the rest of the molecule. So all of this stays the same. The amino group, the carboxylic acid group, the hydrogen, the alpha carbon, and then just this R group gets kind of taken out and plugged back in, like changing out a Lego piece, okay? Or changing out, like if you wanted to turn a Lego from a boat to a car, you just take off some pieces and add some more. That's really what we're talking about with these amino acids. So the 20 amino acids make all the different kind of proteins that we can possibly see, and all of the amino acids have the amino and the carboxylic acid group in common and only vary on the R side chain. So there's just 20 different R side chains that can be plugged in. Once you have the amino acids bind together and form a peptide or a polypeptide. So the amino acids form together into a polypeptide, and the polypeptide folds into a protein. So a polypeptide and a protein are the same thing, they're just folded differently. It's like me saying, like, this piece of paper is a crane. Well, it's not a crane because I haven't folded it into an origami crane yet. Right now it's just a piece of paper, but it could be a crane, or it could be a flower or a butterfly or anything else that I can fold the origami into. Saying that a polypeptide is a protein is kind of true, but it really depends upon how it all folds. So the folding is a really important part of the formation of the protein. Not just the order of the amino acids, but then how are they formed together in order to form their function. So when we talk about how are they folded together, each protein has 4 ascending layers of structure in order to make it into a functional protein. And each protein is going to carefully curate and fold each of these layers so that its final product does the job it was meant to do in the body. We're going to see that even a very small change in even the primary structure could cause a very catastrophic syndrome in the human that has it. So I don't know if you guys have ever seen the Mighty Morphin Power Rangers. Have you guys ever seen it? Any Power Rangers really works. I know now that I have a son that there's also all kinds like Dino Power Rangers and all different kinds. But they all kind of have the same basic plan. And the Power Rangers I'm going to really rely on are the Mighty Morphin Power Rangers, the OG, because I'm old and so deal with it. And so there's a famous Power Ranger, Kimberly. She was the Pink Ranger. Everyone when I was younger wanted to be like Kimberly. She was like so cool, okay? So Kimberly, when she's Kimberly and she's going to school, okay, she is the primary structure. Not really so special. All the potential, but just this like long string of amino acid after amino acid, no real function, no real like fancy, famous, superhero kind of stuff. She's just Kimberly, right? Then at a certain point, Kimberly can morph, right, morphine, into the Pink Ranger. And when she morphs into the Pink Ranger, that's really like her secondary structure, right? She gets some special characteristics. She gets a weapon, she gets some fancier ninja moves, she gets protection from her suit. She's a little bit more specialized in her secondary structure. The secondary structure of a protein is when it folds into something called alpha helix or beta pleated sheets. All of these little spiral staircases and whirls and turns, that's the secondary structure. Once we take all those secondary structures and we kind of add them together, we get something just a little bit greater from the group, and that's the tertiary structure. The tertiary structure is when all of the alpha helices and the beta pleated sheets, they all get pushed together into this big globular protein. That's like when Kimberly goes into her Zord. When Kimberly morphs and she grabs into her Zord and she starts flying that little pterodactyl around, she gets even greater powers, right? Even greater attributes. And that's what happens when a protein folds into its tertiary structure. Kimberly is a fully functional Power Ranger at this point. She can fight battles all alone. And sometimes the tertiary structure is the last structure that a protein has. Other times, a protein is going to do one more structure in order to make it completely functional and effective. This is the quaternary structure. And can you think of what the quaternary structure is from Power Rangers? Well, of course, it's when they form the Megazord, right? When all of the Rangers come together and they make that like big monster thing and it goes down and then it attacks whatever the bad guy is and shoots and kills him. Well, that's what the quaternary structure is. And so the quaternary structure is when all of these globular proteins of tertiary structure proteins combine and form a mega protein. And then they're able to go out and do that specific task or that specific job. Something like hemoglobin in your blood is made of 4 separate tertiary proteins that come together around an iron in order to carry iron and oxygen in your blood. So the structure of our protein is really, really important. And like we've been saying over and over in this chapter, the structure tells you about the function. We just mentioned that hemoglobin took 4 globular tertiary proteins and puts them together in order to carry oxygen in your blood. Well, something that we found is that a single mutation in a single base in your DNA causes a single amino acid out of thousands of amino acids that are in hemoglobin, causes a single amino acid substitution. And that one DNA mutation, one amino acid substitution causes the entirety of sickle cell anemia. So sickle cell anemia is a condition where some of the blood cells are sickle shaped. A sickle is what the Grim Reaper holds. You know the like cane with the like thing? That's called a sickle. And so sickle cell anemia has a sickle shaped cell. And so the structure of the hemoglobin has changed. Well, that also means now that the function of the hemoglobin is going to change. Those sickle-shaped cells are no longer going to be as effective at carrying oxygen. And that's one of the symptoms that a person with sickle cell anemia might have. Another condition is that these sickle-shaped cells tend to hook each other. And so a person with sickle cell anemia not only has a lack of oxygen, but also has a greater risk of stroke and heart attack and blood clot. Sickle cell anemia is not curable, but it is treatable. And at this point in the United States, people with sickle cell can go on to live fairly normal lives, but they have to be monitored closely for blood clots. So as we've been saying, the structure of a protein gives us clues about its function. So I'm going to just look at these 4 proteins with you. The first one is up here. This is actin and myosin. These are proteins in our muscles, and actin and myosin slide past each other to give you your big bicep, right? So if you have like a long arm and then you get like a bicep muscle, that is the actin and myosin that slide past each other, and they contract your muscle, and then the muscle gets beefier, right? Starts to look a little bigger. So actin and myosin are these long, thin filaments because they need to be able to move past each other, pull our bones, and move our skeleton. Down here, we have a protein that allows for transport through the cell. And so you can see that it has this long alpha helix, which is a secondary structure. It has this long helix, and then it has these big proteins on the outside. And so that allows for the protein to be on both sides of the cell membrane and communicate with both the inside and the outside of the cell simultaneously. This over here is an antibody. What we see here is this antibody is a protein, and this light chain can be variable. It can learn. So just like you can learn in a video game, the light chain is able to learn how to fight and deactivate a particular target. And so this is how your body learns immunity. This is what they're all talking about in COVID, and whether or not you have antibodies after the illness in order to prevent future infection. And so these are these antigens attach to a cell, and then they deactivate the cell. The last thing I wanted to look at is the SARS protein spike S. I have had this protein on my slides for 16 years, but this exact SARS protein spike S also exists in COVID-19. And this is the point of attack when we look at forming a vaccine or forming a treatment. We are looking at inactivating this SARS protein spike S as a therapy to help people that have COVID-19. It was also present in SARS, which is a very similar novel coronavirus from the early 2000s. But now COVID-19, which is related to it, a different coronavirus, also turns out to have a similar spike S protein. And so they're able to use some of the information they learned about SARS in order to help with therapies for COVID-19. All right, so these are some specific proteins that have specific structures that help us to understand what their function might be inside our cells. Aren't proteins amazing? I think so.